All right. Hi, Sci 113 students. So welcome to the first speaker series event of the term. Before we get started, I just wanted to let you know we've been asked by the Science Undergraduate Society to promote an event that they have coming up on the sort of early evening of January 20th. This is a coffee chat event where you can sort of learn about basically career options in science. And this, I should mention, is not one of the events that is organized by the Faculty of Science where you can earn participation marks by attending. Rather, this is put on by Science Undergraduate Society, but we thought this might be useful to you, so we just wanted to make sure that you were aware of this. All right, so I'll just leave this up for maybe 10 more seconds if anybody wants to scan the QR code, and then I will hand it over to you, Marcia.
Sure, thanks, Caitlin. And we can leave it up
Oh, sure.
for the introduction. That's totally fine.
Why not?
Welcome, everyone. Yeah, thanks, everyone. It's great to see many of you joining us live. So as Caitlin said, this is our first lecture in the Sci 113 Science and Society speaker series for this term. So just to introduce myself, my name is Dr. Marcia Graves. I'm the director of Sci 113, and I'm also an associate professor of teaching in the Department of Microbiology and Immunology, and I'm really excited for today's seminar. And before I get started to introduce our speaker, before we start, I just want to acknowledge that our speaker series is hosted and that I am joining you today from the UBC Point Grey campus, which is on the unceded traditional and ancestral territory of the Musqueam people. And quite personally, I'm very grateful to be here on these lands. It's been a real place of learning, a real learning journey for me. I was once an undergraduate student, just like yourself, in first-year science, and I've spent much of my life learning here, all the way to being a faculty member at UBC. So it's really been my great privilege. These lands are also a place of learning for Sci 113. This is where a lot of learning happens in the course. And in Sci 113, as you are learning in your first couple of weeks in the course, we explore science as a way of knowing. And through Sci 113, we hope you all grow and gain a broader understanding and openness to how science is practiced and what science is. And in doing so, we recognize and honor Indigenous ways of knowing and Indigenous knowledges, including Indigenous science methodologies, which have long offered important insights into understanding the natural world. So before I introduce today's speaker, to kick off our speaker series, I just wanted to give you all a little bit of context and understanding about this lecture series. We'll have a total of 6 lectures throughout the term, where we've invited a fabulous lineup of guest scientists to come and speak to you all from different scientific disciplines. And the goal is to really hear from a variety of folks, hearing a little bit about their unique journeys in science, hopefully to inspire you, and lots of scientists with different unique perspectives and approaches to how they do science, and just different areas of scientific research. So just a few guidelines for students and a bit of housekeeping. As you know, the lecture series is scheduled throughout the term on Thursdays at 12:30, kind of every other week, pretty much. And for each week, we have a seminar. Just a reminder, there is no class on Friday. So tomorrow, there's no class scheduled. If your schedule permits, we encourage you to join live, as you all have done today. But these sessions are being recorded, so the links are provided for you on the Canvas site for section 201. And I wanted to just reiterate that it's important to keep up with the talks. So even if you aren't able to join live, do watch the recording so that you're prepared to engage in discussion about the lecture in your next class, so on Monday. And also the scientific topics that are discussed throughout the term will be relevant for your final project. So it's really important, as I say, to make sure that you're engaging in all of the talks across the term. As you know, there's also a worksheet associated with each of the lectures given throughout the speaker series, and you're expected to complete your choice of 4 out of 6 worksheets for the term, okay? And the worksheet for today's talk is due on Saturday. So during the talk, definitely we encourage you to think of questions you'd like to ask the speaker. And for those of you joining live, there'll be time at the end to ask questions in the chat, or you may raise your hand in Zoom, and we'll try and address each question. Okay, without further ado, it's my absolute pleasure to introduce our first speaker of the speaker series for this term, Dr. Mark MacLachlan. Dr. MacLachlan grew up in Quesnel, BC, and completed his undergraduate degree here at UBC in honors chemistry. After completing his PhD, he moved on to the University of Toronto, he completed his PhD at the University of Toronto, and a postdoc at MIT. He returned to UBC as an assistant professor in 2001. Dr. MacLachlan has taught Chemistry 121 many times, so many of you may have taken or are taking that course, and has taught several other courses in inorganic materials in chemistry for undergraduate and graduate students. He leads a research group in chemistry that's focused on topics in inorganic and materials chemistry that range from constructing molecular rings and knots to developing new materials that mimic the structures and iridescence of beetle shells. So I think we'll hear about that today. Dr. MacLachlan holds the Canada Research Chair in Supramolecular Materials and is a Fellow of the Royal Society of Canada. Dr. MacLachlan served as Associate Dean for Research and Graduate Studies in the Faculty of Science here at UBC, and is in fact now serving as our Dean Pro Tem for the Faculty of Science. So it's really an honor to have you join us today, Dr. MacLachlan, and I'm really looking forward to your talk.
Just as we get started, I'm seeing the presentation in presenter view rather than in the full screen view. Would we want to try again with the screen share? Maybe.
Okay, let me try that again. So, is that the right view?
Perfect. Yeah, this time it's
Looks great.
Okay, awesome. Thank you so much for the introduction, Marcia. And it's great to see you all, or to at least see your names listed on the Zoom page. I'm so delighted to be the first speaker in this series and have a chance to tell you about some of our research from here at UBC. So, as Dr. Graves mentioned, I grew up in the center of British Columbia in a little town called Quesnel. That's where I did all of my public schooling. And then I came to UBC as an undergraduate student and completed my Bachelor of Science in 1995. Following that, I went and did a PhD degree in Toronto, worked at MIT as a postdoc for a couple of years, and then I was fortunate enough to get a job back at UBC starting in 2001. And since 2011, I've been a full professor in the chemistry department at UBC. So what does a professor do? I guess there's three things that we do primarily: teaching, service, and research. So in terms of teaching, we teach undergraduate and graduate courses. We prepare for lectures, give lectures, hold office hours, change curricula, prepare exams, and do marking of exams. In terms of service, a lot of us serve on different department committees and university committees for things like scholarships and facilities. We review grant proposals and research papers and write a lot of letters of reference for students who are pursuing graduate school or professional school. For research, we usually supervise master's, undergraduate, and PhD students on research projects. We write research papers, write grant proposals to raise money to pay the students and pay for our equipment, travel to give talks and attend conferences, and write reports. Now, as Dr. Graves mentioned, I've been serving as the Dean of Science Pro Tem for just over a year, and probably you have no idea what a dean does. So I thought I would just highlight kind of what that job entails. So it's a full-time role of administration. In this role, I oversee the budget for the entire Faculty of Science, the hiring and management of 12 departments and institutes, and we have 4 museums in the Faculty of Science. Provide oversight of the academic programs and student advising. I'm heavily involved in the recruitment and registration of students into first year. Oversee graduate programs in science, research administration, research funding, and often I represent faculty at different events and serve on committees. Luckily, I still have some time to do research, and so it's awesome that I can today tell you about some of the research that we've been doing at UBC. So I'm going to shift gears completely, and here's a picture of a bone. So bone is a very lightweight, hard material, and it has amazing properties. If you just think about what your bones go through when you're jumping and running for, it might be up to almost 100 years of your life, these bones go through an incredible amount of situations. And the reason that they have such amazing properties and strength is because they have what we call a hierarchical structure. So at the nanoscale, they have small hydroxyapatite bone crystals that are embedded with collagen molecules. These are organized into fibrils, which are further organized into collagen fibers and into a lamellae, and then ultimately into osteons that form the basis of the bone. So there are different structures that span numerous length scales ranging from nanoscale to centimeters. And this is a common theme in nature, that materials get their strength and amazing properties because they have hierarchical structures. Some other examples of amazing materials include spider silk, which is the strongest. It's much stronger than steel for its weight, and that arises because of its hierarchical structure. Gecko feet. Geckos are able to hold enormous weight and walk upside down on ceilings because they have this amazing nanostructure on the surface of their pads that lead to an extremely high contact area and strong van der Waals interactions between the gecko and the surface it's walking on. So my research is in a field called supramolecular materials science. Supramolecular means beyond the molecule. So just as you might think about assembling Lego into complex structures, we think about taking molecules, polymers, and other building blocks and using non-covalent interactions such as hydrogen bonding, dipole-dipole interactions, and so on to organize them into much more complex structures. So ultimately, we hope that we could take molecules off the shelf and coerce them to self-assemble into a structure that's as complex as an individual cell. Plants also have amazing hierarchical structures, and some of these structures lead to the mechanical properties of the plant and also to the colors. And so I want to spend a little bit of time talking about the color of materials and talking about light as a refresher. So colors, for those of us who are able to see them, are beautiful and add an incredible amount of spice to our lives. It was Isaac Newton who in about 1670 discovered that white light is made up of a spectrum of the colors. So when he passed white light, which was just sunlight, through a prism, he was able to split the light into all of its native colors, ranging from red all the way to purple. So if you think of light as a wave, which I'm sure all of you have learned, then one of the key properties of the light is its wavelength, and the wavelength correlates with the energy of the wave as well. So long wavelength light, which is low energy, at around 700 nanometers corresponds to red light, and as we decrease the wavelength down to about 400 nanometers, we can cover all the visible colors ending at blue or purple. Excuse me. So the visible part of the spectrum is actually just a really small, practically insignificant part of the total electromagnetic spectrum, which spans from the longest wavelengths of radio waves to the shortest and highest energy waves of gamma rays. So visible light is this very small but important to us because we are able to sense visible light very well with our eyes. Now, when you look at an object, like I'm wearing a colorful shirt today, then the colors that you see arise from a property called absorption. So there are chromophores or molecules in my shirt or in most objects that absorb light. Light is absorbed to allow electrons to undergo a transition from a ground state energy to some excited state energy, which we call molecular orbitals. If you look at a green plant, it is absorbing light around 400 to 450 nanometers and also at around 650 nanometers, red light. The light it doesn't absorb is the green light in the middle. And as a consequence, it is the green light that gets reflected from plants when you shine white light on them. And this is why you see all plants look green, because of the absorption of chlorophyll. Other sources of color exist. For example, interference due to an oil slick on a water surface. So incident light undergoes reflection from the air-oil interface, but it can also undergo reflection from the oil-water interface. And because of the oil has a different refractive index than the air, these two waves will be out of phase and undergo constructive or destructive interference. And that leads them to give this impressive colored pattern that you see due to interference. And finally, another possible source of color is due to a property called diffraction. And I'm going to talk a lot more about this in this talk. If you have a periodic structure, like a crystal, then the waves can be diffracted from the different planes. And at a certain angle, they will undergo constructive interference, and that particular wavelength will be selectively diffracted from the color. And some of you may have learned Bragg's law, which tells us the relationship between this angle theta and the color of the wave, or the wavelength that is reflected from the periodic structure. So now that I've reviewed light and the origin of color in our world, I want to tell you about something really boring, and that is paper. So probably most of you have already had an encounter with paper, even today. But I doubt that many of you have looked at paper under an electron microscope. But if you do look at it under an electron microscope, you'll see it's got this woven fiber structure. And those fibers are made of a substance called cellulose. Cellulose is the most abundant biopolymer on Earth. And it has a chemical structure, which is shown on the top right here. These are glucose molecules, which are linked into long chains. So glucose is a regular sugar that we eat all the time, and it fuels our bodies. And when you link these into long chains, you can form starches or cellulose. When these chains of cellulose then are hydrogen bonded to form crystalline regions of cellulose. Now, in a piece of paper, there are both crystalline or organized regions of cellulose and amorphous or disordered regions of cellulose. But it was shown in the 1950s that if you take a piece of cotton or a piece of paper, which are mostly made of cellulose, and you treat it with sulfuric acid under the right conditions, then you can selectively hydrolyze away the amorphous regions and isolate these nanocrystals of cellulose. This is an electron micrograph of cellulose nanocrystals, and I'll show you that up close in a moment. An important part of this process is that the cellulose nanocrystals get coated with these sulfate groups, which give them a surface charge and allow them to form stable colloids in water. So this is that electron microscope image of the cellulose nanocrystals. These cellulose nanocrystals are about 5 or 10 nanometers in diameter and maybe around 200 nanometers long. We have to use an electron microscope to image them because they are smaller than the wavelength of visible light. You can't see them with a regular microscope. And just to give you some context, you would have to put about 1,000 of these end to end to be the width of a single hair. So these are true nanomaterials. Now, people around the globe are exploring cellulose nanocrystals for a lot of different applications that range from paints and adding them to ice cream to change the viscosity. But I want to show you something really cool that we are exploring with these cellulose nanocrystals. They have an amazing property. On the left is a beaker of dilute cellulose nanocrystals in water. And as you might expect, they're colorless. But if you let the water evaporate from this beaker and concentrate those cellulose nanocrystals, you get a colored iridescent film, as shown on the right. So there is no dye or pigment being added, but we're getting color from a material that is colorless. And the origin of this color is something called structural color. Now, structural color is found in many examples in nature. There are reptile skins, fish scales, and insect shells, for example, that get their coloration from structural order at the nanoscale rather than pigmentation. Birds have many examples of structural color. If you see blue bird feathers in nature, then you can be sure that's due to structural color because birds have not evolved blue pigments for their structural color, for their coloration. There are even some berries that get their coloration from structural order at the nanoscale rather than pigments, and fossils and minerals as well. Now, the most famous example of structural color is the morpho butterfly. Shown here is this morpho butterfly with these beautiful blue iridescent wings. But in fact, there's no blue pigment in these wings. If you hold it up to a bright light, the wing just looks a pale brown color. If you look at the surface of the wing, it has these features that look a little bit like trees, and the branches are about 400 nanometers apart. And that distance matches blue, the wavelength of blue light. So when white light is incident on this wing, blue light undergoes constructive interference and is diffracted from the surface of the wing, making it look iridescent. So coming back to cellulose nanocrystals, we get a colorless film, which exhibits structural color, from a colorless substance. And in this case, it's because the cellulose nanocrystals organize at the nanoscale. And they organize into something called a chiral-nematic order. So I want to illustrate for you what this looks like. Imagine that these rods, the cellulose nanocrystals, they tend to align into layers as they dry. So the first layer, all the cellulose nanocrystals are kind of aligned parallel to one another. In the next layer, they're also aligned parallel to one another, but twisted a little bit relative to the first layer. The third layer, they're aligned, but again, twisted a little bit farther. And so what you build up in a film of cellulose nanocrystals is a series of layers where you have the cellulose nanocrystals aligned in layers whose orientation rotates through the film. And when the pitch of that rotation matches the wavelength of incident light, that light gets diffracted. So on the right here is a depiction of the orientation of the cellulose nanocrystals in the layers. And when the helical pitch of that structure matches the wavelength of incident light, it gets reflected. And this is called iridescence. Now that I've introduced you to the cellulose nanocrystals and their organization into a chiral-nematic order, I want to talk about our research. So we wondered if we could capture this chiral-nematic organization of cellulose nanocrystals in another material like glass, or what we call silica. So there's a way to make silica, which is just this, it is the glass that we make all of our windows out of, and it's also sand. There's a way to make silica from a simple molecule called tetramethoxysilane. If you take tetramethoxysilane and put it into water, it will undergo hydrolysis and condensation to give silica, SiO2. And silica is a three-dimensional network of tetrahedral silicon atoms bridged by oxygens. And when you do this at room temperature, the silica you get is amorphous. It doesn't have crystalline order. So here's our chemistry. We take our beaker of cellulose nanocrystals in water, and we add tetramethoxysilane, that glass precursor, and then we stir it. And then we pour it into a petri dish. So this is a petri dish containing cellulose nanocrystals, tetramethoxysilane, and water. All we do is leave this on the bench top and let the water evaporate. So I'm going to show you a video of the water evaporating from this petri dish overnight. So watch closely. Initially, it's completely colorless. But you can see the color magically appear as those cellulose nanocrystals organize into this chiral pneumatic structure. But at the same time that the cellulose nanocrystals are organizing, that molecule tetramethoxysilane is condensing to give glass. We now have a film that's about half glass and half cellulose. And you can see the iridescent color, which tells us that the cellulose is organized into that chiral pneumatic structure. So we've got a mixture of cellulose and silica. What do we do next? Well, we take this film and we burn it. So we cook it at 500 to 600 degrees Celsius under air. That destroys all the cellulose, converts it into water and CO2, and leaves us with pure silica, pure glass. So the materials I'm going to tell you about now are just made of silica. All the organic, all the cellulose nanocrystals have been removed. So this film is very porous. We use a technique called gas adsorption measurements, which are done at liquid nitrogen temperature. And this gives us information about the pore size distribution in the film. And what we find is that the films have a surface area of about 650 meters squared per gram. And to give you context, 1 gram of this material has half the surface area of an NHL ice rink. So it's got a very high surface area inside of its pores. We can get the pore diameter, and we find that the pores are around 4 or 5 nanometers in diameter, which is pretty similar to the diameter of the cellulose nanocrystals that we use as a template. Now, silica is a colorless substance. That's why we make all of our glass windows out of silica. But our materials are colored, and we can make films that are blue, green, red, yellow. We can make films that are colorless but reflect infrared light, or films that are colorless and reflect UV light. We can tune it by about 2,000 nanometers, including all across the visible spectrum. Now, because the color originates from a diffraction property, there's a strong angle dependence on the color. So if you look at the yellow film from the top and you just tilt your viewing angle, it turns blue. So there's actually a sine theta angle dependence on the wavelength of light that is reflected from these materials. Now, to study these materials, we look at them by a lot of electron microscopy, in particular scanning electron microscopy. If you take a film and cut it and look at the cross section, you can see a pretty well-ordered layered structure. The top of the film is relatively smooth, and the edges have these undulating features. We've looked at these by high-resolution electron microscopy, and then you can actually resolve a left-handed helical twist to the silica. So a reminder that there's no cellulose here. This is the pure silica film that we're looking at, and we can see that the helical structure of the cellulose nanocrystals has been transferred to the glass through this templating effect. So what we've been able to do is to use cellulose nanocrystals as a template for chiral pneumatic organization. The solid material that we get after using cellulose nanocrystals template is the negative of it. It's a solid with rod-shaped pores that are organized into this helical structure. So what can we do with this material? Well, one interesting thing is that it's very porous. So if you take a film, in this case the green film, and you add a drop of water to the surface, then the water rapidly goes inside of the channels and it becomes colorless and transparent anywhere that the water has infiltrated, because the refractive index of water is pretty close to the refractive index of glass. Here's a photograph of those 4 samples with droplets of water. And if you dry them, then it goes back to the original. In fact, because this is structural color, these materials can keep their color even if you heat them at 500 degrees in the air for days. They will not lose their color. They will never fade. I've had samples of these on my desk for over 10 years, and they look exactly the same as when we made them. You could shine lasers on them, and because they don't absorb light, they only reflect it, they won't undergo any optical damage. So there's lots of interesting applications for these materials. Now, one thing you'll note is that we often have a coin on these photos, and that coin is there to give you a sense of the size. So this is a Canadian dime, which is our smallest coin. And for a long time, these were the smallest or the largest pieces that we could make. When we made these films, they always cracked and shattered, and we couldn't make very large ones. But we made an accidental discovery several years ago that has allowed us to make much larger films without cracking. And the problem is that as the films dry, there are pressure gradients that develop and lead to the cracking. So what we discovered was that if you add sugar to the sample before it dries, then you can completely eliminate the cracking. And these are shown samples with no sugar added, 8 weight percent, 16, and 28 weight percent. So after 28 weight percent, we get a large crack-free film. Here now is a photograph of that composite of cellulose nanocrystals and silica made using 28 weight percent glucose beside the dime. So again, we can make much larger films that are crack-free. When you calcine this in air, we can make large crack-free films of the pure silica, and you can see that it's iridescent again. Here you can see the photograph of the red film from the top, and if you tilt your viewing angle, then again it turns blue or green because there's this sine theta angle dependence on the color. If you add water to the left side of the film, then it again goes inside of the pores and makes it colorless and transparent in that region, and it's completely reversible. We can use these for humidity samples, sensors, for instance. I want to show you something really interesting. This is a pair of 3D glasses. If you go to a 3D movie, the way that they work is they generate 2 polarizations of light. They produce one polarization of light that is what we call left-handed circularly polarized, and one polarization of light that is right-handed circularly polarized. So in the 3D movie, there are 2 projectors, one that's projecting an image with left-handed circularly polarized light, and one that's projecting an image with right-handed circularly polarized light. You wear these 3D glasses, which have filters for left and right-handed circularly polarized light. So I want to show you what our materials look like if you wear 3D glasses. This is a sample of chiral pneumatic mesoporous silica. And through one lens, it looks perfectly normal, but it completely disappears through the other lens. And what this is showing is a really interesting physical property of our materials. Because they have a left-handed helical structure, they only reflect left-handed circularly polarized light. So we're exploring some security applications where you could embed these materials or similar materials into a passport or into other important documents. And a person could verify with a pair of 3D glasses whether it had the right helicity And this is very difficult to reproduce in your kitchen or in your basement if you're trying to make some fake documents. So some of the potential applications of this material that we're exploring, we're looking at separation of molecules. Many molecules exist in a left-handed or a right-handed form called enantiomers. And it turns out that they travel at different rates through our material because our material has a left-handed helical twist. And so we can use these silica films to separate these kind of molecules, which are otherwise extremely difficult to separate. We're looking at them for color filters and polarizers, reflectors of infrared light to reduce the amount of heat energy that gets into buildings, sensors, where responding to some kind of guest binding inside of the pores changes the color of the material, templates for other materials, catalysts, and decoration. We've been exploring making ornaments and window coatings or enhancing the color of paints using these kind of materials. Now, I started off talking about nature, and you're probably wondering what the connection with these materials beyond the fact that they're made using cellulose nanocrystals, a natural material, whether there's another connection to nature. And it turns out that this beetle, called the jewel beetle, native to many parts of Asia, it has these beautiful iridescent exoskeletons. And the iridescence comes from the fact that on the surface of this structure, it has a chiral-pneumatic organization of chitin nanocrystals. So this beetle has evolved the way to make a left-handed chiral-pneumatic ordered structure of chitin nanocrystals on the surface to give it iridescence. So our materials are kind of a mimic of the jewel beetle. Now, those materials I've been talking about are very brittle. If you handle this glass, it's extremely delicate material. It cracks really easily. So we've spent a lot of time trying to extend this concept to other more flexible materials like plastics and hydrogels. So I'm going to tell you a little bit about a couple of materials we've made. Now, hydrogels are materials that are cross-linked hydrophilic polymers. That is, they're networks that love water, but they're also linked together so the polymers can't dissolve. So this leads them to be super absorbent. And you've all encountered hydrogels before, whether in baby diapers or contact lenses or these swellable toys. And there's lots and lots of other applications for hydrogels. So we wanted to make a hydrogel that had this chiral-pneumatic organization and could be colored. So what we imagined doing was taking cellulose nanocrystal suspension and adding chemicals to it so that we could polymerize and form a hydrogel around the chiral-pneumatic organization of the cellulose nanocrystals. And so this is what we did. And you don't have to worry too much about these molecules, but we took a common precursor to polyacrylamide, which is a well-known hydrogel. We took acrylamide and this molecule on the bottom, which is a cross-linking agent, and we combined them in water with cellulose nanocrystals and a free radical initiator for polymerization. And then we watch it as it dries under a polarized optical microscope. And once the cellulose nanocrystals start to form this organization, then you can see this fingerprint texture arise. And so then we know it's organizing. We irradiate it with UV light to initiate polymerization, and we capture the chiral-pneumatic organization of cellulose nanocrystals in a hydrogel. Shown here are two hydrogels we prepared. On the left is one where the cellulose nanocrystals have a helical pitch that is longer than the wavelength of visible light, so it's reflecting infrared light. And on the right is one that is reflecting visible light because the helical pitch of the cellulose nanocrystals is on the order of the wavelength of green light. Now, as I mentioned, hydrogels swell in water, but they don't swell in alcohol or ethanol. So what we explored was using these as sensors for detecting the concentration of ethanol in water. And if you take the hydrogel, initially it reflects light around 500, these are reflection spectra for the samples. Initially, it reflects light around 550 nanometers. And that's if you put it into pure ethanol, it doesn't swell at all. It still reflects light around 550 nanometers. But if you put it into liquids that contain increasing amounts of water, then you can see that the reflection redshifts across the spectrum. And so to our naked eye, we can detect the color of the film, which correlates with a particular concentration of alcohol in the water. And we can use these for detecting, or for reversibly sensing the concentration of alcohol. And just to let you know, this ethanol, that is the alcohol in vodka and other alcohols that people drink. We extended this concept to other hydrogels. We can make polyacrylic acid hydrogels, and now the color of the film is determined by the pH of the solution. And again, we can just dip our hydrogel into the solution, and by measuring or observing the color change, we can tell what the pH is. So this is a very expensive pH sensor. We can also make them with poly-N-isopropyl acrylamide, which is a temperature-sensitive hydrogel that allows us to measure temperatures very close to the body temperature. Now I showed you this hydrogel, which is dry and reflecting green light. If you put this particular sample into water, then it looks like this. So we are able to photo pattern these films so that different parts of it will swell to a different extent and present a different color. You could imagine if this is your shower curtain, and when you turn the water on in your shower, suddenly you'll get a pattern, and we could make multi-color patterns for very nice shower curtains. And so we're exploring some of these kind of decorative applications as well. Another goal that we've had for a long time is to make elastomeric materials with this structural color from cellulose nanocrystals. But for a long time, we couldn't overcome a technical barrier, which is that cellulose nanocrystals are hydrophilic. They love water. But elastomers, like the materials in rubber bands and other plastics, and their monomers are all hydrophobic. They phase separate out of water. So we spent many years trying to make elastomers with cellulose nanocrystals, but failed miserably in our lab. But then I had a brilliant visitor from Japan in my lab, and he found a way to overcome this. So I'm going to walk you through the process that we use to make elastomers in our lab with chiral pneumatic organization. So the starting point is and this is important that we add glucose. We start with a solution of glucose and cellulose nanocrystals in water, and then we allow it to dry. And we call that evaporation-induced self-assembly. So all you do is take that mixture of glucose and cellulose nanocrystals, leave it to dry, and you get a colored iridescent film because the cellulose nanocrystals have organized into this helical structure. The next thing that we do is we soak it in a solvent called dimethyl sulfoxide. Here's the film before soaking, and we swell it in dimethyl sulfoxide. Now it's transparent and colorless because the helical pitch is longer than the wavelength of visible light. So initially it was reflecting visible light, but when we swell it, it's elongated the helical pitch so that it's now reflecting infrared light. Now what we can do is we can take this swollen film and we can immerse it in a mixture of monomers. And we spent a long time optimizing this, and what we found works well is a mixture of 2-hydroxyethyl acrylate and ethyl acrylate. These are commercially available monomers. We can buy them by the bucket load. We swell it with those. We add an initiator, a thermal initiator in this case called AIDN. Again, just something that we buy. And then we heat the film at 60 degrees for 18 hours. And what we get is a colorless, transparent film. And this is a mixture of cellulose nanocrystals and our elastomer. And if you take a cross section of this film and look at it under an electron microscope, then you can again see the well-ordered layered structure that emerges from the chiral nematic organization of the cellulose nanocrystals. As I mentioned, this is an elastomer. So when it's relaxed, you can see it's colorless and transparent. And when you stretch it, it remains colorless and transparent, but we can stretch it up to 8 times its original length, and it's completely reversible. This is a beautiful elastomeric material. Unfortunately, for the longest time, we could not get structural color. Then I had a brilliant postdoc, Charlotte, and a great graduate student, Andy, who worked together and optimized the conditions for this process so that we could transition it into the visible part of the spectrum. So they optimized each of these steps over about a year until they were able to make what I think is a really amazing material. So this is a photograph of the elastomer cellulose nanocrystals composite that they produced. And importantly, it has a chiral nematic organization of cellulose nanocrystals with the helical axis coming out of the screen towards you. So I'm going to show you what happens when you stretch this material. So initially it's completely colorless, but as you stretch it, it becomes colored. And we were able to prove that the reason it becomes colored is because as you stretch it, the helical pitch shrinks from the infrared region into the visible part of the spectrum. And we can use these as pressure sensors or stress sensors because their color depends on the amount of elongation. So this is a photograph where we have the unstretched material. You can stretch it to become red, green, or blue. And then we use reflection spectroscopy to measure and quantify the amount of reflected light that comes from the surface of these. One neat thing with this process is that our starting glucose cellulose nanocrystal film can be patterned. So we've got some cute tricks to pattern these films. In this case, we made a patterned film where the right side has a helical pitch corresponding to red light, and the left side has a helical pitch corresponding closer to green light. We can also pattern our films by adding droplets of water, and that changes the helical pitch where the water's been added. So we made patterned films with the elastomer, and they are colorless and transparent. But when you stretch them, then you can see a pattern emerge. So in the top right, you can see the red spots where the water droplets were added to pattern it inside of a blue background. On the bottom, when you stretch it, you get a gradient of colors perpendicular to the stretching direction because of the way that we patterned this film to begin. So we are thinking about being able to pattern, for example, a company logo or maybe UBC logo onto these materials so that you don't see it at all until you stretch the material and suddenly it would present this colorful pattern. So that is basically all the science I'm going to talk about today, but I want to tell you a little bit of a story about us publishing and give you a little bit of insight into how we publish this. So this last part of the story, the photonic elastomers, I think is one of the most exciting things that we've done in the last 10 years. And we were really excited about this. So at some point, we decided we had enough results that we had a story to tell. So I told Charlotte and Andy, let's write a paper and submit it for publication. So Charlotte and Andy worked together over a period of a couple of months and wrote a draft of a paper, which includes an introduction, a long discussion about our results, lots of graphics, and an experimental section, just like a lab report. And we sent it back and forth between the three of us for editing about 5 to 10 times. So they would work on it They would send it to me. I would spend 4 or 6 hours going through the manuscript. I would make edits, I'd make comments, asking them to address certain things, and then I'd send it back to them. And we go back and forth until we think that we have a near perfect document that we're ready to submit for publication. So we selected a journal called Advanced Materials, which is one of the very top journals in material science. And we submitted this on July the 3rd, 2019. Then we wait. So what happens is you submit a paper to a journal, and then they send it out to peer reviewers around the world to evaluate the quality and novelty of the science. And in the field of chemistry, we typically get responses in 1 to 2 months, whereas some other fields like math and geology, it's not unusual to have to wait a year for the peer reviews to come back. So we submitted it on July the 3rd, and by the end of August, we got the initial review. And I'm going to share that with you. Dear Professor MacLachlan, my apologies for the negative decision. Thank you for submitting your manuscript. The reviewer report and comments are included at the end of this email. Unfortunately, on the basis of the reviewer comments, we are not able to accept it for publication. We therefore recommend you submit your manuscript to another journal, and we hope that the reviewer comments will be useful in revising your manuscript. So we regret disappointing you on this occasion. I'm not so sure they really regret it, because I think they do it a lot, but nonetheless, they wish us success in publishing our work in another journal. So I'll show you the review. It turns out that we only got 1 peer review for this paper, and that was the basis for the editor's decision. And this reviewer had to answer a few different questions about the paper. So please rate the importance of this submission. This reviewer said it should be submitted somewhere else. Please rate the originality of this submission. They said it's got an insufficient level of new results. Please rate the scientific and technical content. They were happy with that. It was fully consistent and accurate. And the length was fine. It's concise and correct length. But really important for this kind of journal to have sufficient novelty and originality for publication. But then there were some comments from the referee, and I'm not going to read all of these, but I'll just say that their point was that this work is only incremental in terms of novelty compared to some previous work that we had published, which was the elastomers that didn't change color when you stretched them, and it does not fulfill the novelty criteria required by a publication in Advanced Materials. Publications in a more specialized journal is recommended. However, we were really disappointed and thought this work deserved to be published in this top journal, and we were annoyed that there was only 1 reviewer. It's a bit of a crapshoot, the reviews that you get. Often you'll get multiple reviewers with completely different opinions about the same paper. So I appealed this decision back to Advanced Materials and asked them to reconsider their decision and send it out for additional peer review. And they got back to me pretty quickly. Dear Mark, thank you for your arguments. I am afraid I would agree with the referee concerning the novelty of your current submission. On the other hand, I've checked with my colleague working with Advanced Optical Materials, which is a significantly inferior journal compared to Advanced Materials, and they would be happy to consider a transfer of this paper to AOM. Andy and Charlotte were not satisfied with going to this journal. They thought the work was better and deserved to be published in a better journal. So we went on to the next journal. We submitted the paper and this requires us to revise the paper again because every journal has a different formatting, different structure, but it takes us maybe a week to get it reformatted. And we submitted to Angewandte Chemie, which is a top chemistry journal, on September the 7th, 2019. And just 2 weeks later, on September the 22nd, we got our reviews back and comments from the editor. And this time, the editor says, our impression is that the above-mentioned manuscript might indeed become suitable for publication in Angewandte Chemie. However, the comments and suggestions from the referees requires that we defer our final decision until you have considered their remarks and revised your manuscript accordingly. So pretty positive. This time there were 2 referees, and by the way, I don't know why previously it was just reviewer number 2, because there was no reviewer number 1. I suspect that they sent it out to 3 or 4 referees and only got 1 back, which happened to be the one labeled reviewer number 2. This time they got 2 reviewer reports, reviewer number 1 and reviewer number 2. Number 1 said our paper was in the top 5% of papers published in this journal, and the second one said it was highly important, top 20%. Both of them said that we didn't appropriately cite previous publications, and I'll come to that in a moment. The length, one said it was concise, the other one said the paper was too short. And in terms of verifying the hypothesis and conclusions presented by the data, fully consistent and minor inconsistencies. So I'll show you some of the details of the reports. So referee report number 1, I'm not going to read it all out, but they said that they think that the paper would benefit from a broader overview of cellulose nanocrystal based composites, especially ones involving optical properties. And so this was their point about the additional citations. They asked us to add a couple of different references, and we did so in revising the paper. So otherwise, I'm happy to suggest this paper is published. So that was a very positive referee report. Referee report number 2, and by the way, these referees could be anywhere in the world. They're almost always professors, but they could be people working in industry. And you never know who they are because it's anonymous peer review. So referee report number 2, they had some problems with the paper that is scientifically, it's a rather minor development from the paper that the team of the senior author recently published in Nature Communications, and that was the one I alluded to before, which was where you can stretch it and they don't change color. They employ the exact same materials following qualitatively the exact same procedure and obtaining qualitatively the exact same structure. And so they suggested, though, some changes that would make this appropriate for Angevin chimie. So this is the first page of their referee report. The next page, they gave us some suggestions about improving the paper, and then they started asking questions. So question number 1, they wanted more information about the role of the glucose and for us to carry out some experiments to figure out how it was interacting with the cellulose nanocrystals. And then point number 2, they wanted to know information about how the cellulose nanocrystals were connected with the elastomer network, and they had a number of points. And this review went on for many pages. So after reading these referee reports, it's required by us that we make a point-by-point rebuttal of all these points of the referees, and that we revise the manuscript so that the referees are satisfied and that it's suitable for publication. So we did that. We worked on the paper for quite a while, made the revisions, and then when we sent it back, it went back to the referees and they were satisfied with the revised manuscript. And so it got published early in 2020, just before the onset of COVID. And this is what the paper looks like. This is the first page. I think it's a 6-page paper. And it got what we call very important paper status because one of the referees indicated that it was in the top 5% of the papers that they publish in Angevin chimie. So just to recap, we've developed many new materials with tunable chiral-pneumatic structures. We can make glass, plastics, hydrogels, elastomers, and so on. I think a lot of these materials are promising for sensors, imprintable photonics, window coatings, and decoration. And they mimic the hierarchical structures of beetle shells. Now, one important thing I have to say is that I did not do any of this work myself. In fact, I haven't done a lab experiment in our research labs for about 15 years. I have an amazing group of co-workers, which includes undergraduate students, PhD students, visiting scientists from other countries, and postdocs who contribute to this work. And I'll just mention a few of the undergraduate students. Bowen is an undergraduate student. Kosei was an undergraduate student in our group last year. He did his 4th year project in our lab, and now he's a PhD student. Rihanna is an undergraduate student. Joe and Carl and Callie and Ying Hao are all undergraduate students who work on projects in our lab. They do real experiments. They're not in there just washing glassware. They all have designated projects, and they work closely with a PhD student or a postdoc. I need to thank all these different organizations that fund various parts of our research. As a PhD student, you get paid, and it's up to me to raise the money to keep paying students and paying for chemicals and instrument time and so on. So once again, thank you so much for having me. I really enjoyed every opportunity I can to talk about our research, and I'd be happy to answer any questions you might have.
Amazing. Thank you for that, Dr. MacLachlan. Great seminar, great way to kick off the term. So at this stage, I invite all our students in the audience to share your questions for Dr. MacLachlan, either in the chat, or please feel free to raise your hand in Zoom, and we'll try and address everyone's questions. And just a reminder to keep things respectful in the chat as well. Maybe we'll start, Mark, I don't know if you can see all the questions in the chat, but I'm happy to read them out if that's helpful, or...
Sure, if you could read them. I do see a couple participants have raised their hands too.
Sure, we'll just start with, I saw Daniel in the chat first, and then we'll come to Anthony, who's got their hand raised as well. So Daniel has a question in the chat. What inspired you to pursue the research of iridescence? Is there a particular moment in your life or experience that was a driving force, or was this kind of a spontaneous thought and it came to fruition? Great question.
That is a great question. And I wish I had told the story of how this happened. So my research was making ring-shaped molecules that can be stacked into nanotubes. And it's a lot of organic chemistry, some coordination chemistry. And there was a person who was coming and using some of our analysis equipment in our lab. His name was Wadud, and he worked at FP Innovations, a company that works on forest products. I had no idea what he was doing in our lab, but I was charging him to use the equipment and making some money from it, which was great. One day he came to me and he said, "We're working with this material called cellulose nanocrystals. Do you want to do something with it?" And I actually ignored him for probably 6 months or a year. I didn't want to get involved because I was too busy. And then one day he came to me and he said, "Mark, I want you to work with cellulose nanocrystals, and I'll give you some research funding." So I couldn't turn that down. And what we did was we proposed to use cellulose nanocrystals to make aerogels that could be used for storing hydrogen in hydrogen-powered cars. So the idea was to have cellulose nanocrystals randomly organized in silica, in a glass, and then burn them out and have a porous material that could absorb hydrogen on board of a car. And I had a PhD student who made hundreds of samples and measured them. But one day, this student, Kevin, came to me and he said, "I don't know what I've done, but the sample is colored." He had made hundreds of samples that were all colorless, but this one sample was colored. And we thought that was something pretty interesting and surprising, and he was able to reproduce it. And it turns out, I did not know anything about structural color before that. I didn't know that cellulose nanocrystals formed this chiral pneumatic structure, and it completely transformed my research career. So we've now published about 150 papers with cellulose nanocrystals, and I got into it completely by accident. So that's a great question. Thank you for that, Daniel.
Yeah, it's amazing how there can be those happy accidents, those surprises in science.
Serendipity.
Yeah, absolutely. Let's go to Anthony. Go ahead. You've got your hand raised.
I was wondering how the undergraduate students got to be a part of the project.
Yeah, good question. So I've had about 200 undergraduate students work in my lab over the last 20 years. And I always have a number of undergraduate students. Some of these students, like Joe, is volunteered in the summer and she has continued to stay on. Same with Rihanna. Carl is doing his Chemistry 449 project, which is the honors thesis project in our lab. Callie is doing a Chemistry 445 individual research project in our lab. Ying Hao is actually a physics student. He's doing a physics 449 project, but he worked in our lab for the summer and is now analyzing some of our materials over in the physics department, but he spends most of his time in our lab. So there are lots of opportunities through co-op, through volunteering, through summer scholarships. We have a summer scholarship program called the NSERC USRA Undergraduate Summer Research Award. And also there are research courses, like I said, Chem 449, Chem 445, and every unit across the campus has analogous research courses. So there's lots of opportunities to get into research, starting as early as first year, but many students, I didn't start until after my third year. I do encourage you to get a research experience after second, third year if you can.
Yeah, great. That kind of addresses Hajar's question in the chat, which was precisely Anthony's, like, how can undergraduate students be part of scientific research groups? And so what you suggest through a co-op or some of these research opportunity courses like directed studies and things like that. Do you have any other advice, Dr. MacLachlan, for students interested in getting involved in research?
Yeah, I think one important thing is to go to your professor's office hours. If you're interested in chemistry, for example, and you're taking Chem 123 now, go to that professor's office hours, connect with them. Because then if you email them or talk to them and ask them if there's any volunteer opportunities or opportunities to engage in research, they'll know who you are and be more likely to find an opportunity to get you in the lab. When I taught Chem 121, I used to always pick a few students that were at the top, near the top of the class, and invite them to be part of our research lab in the summer after their first year. And had some great students go through our lab as a result.
Yeah, that's awesome. I would also add to chat with your teaching assistants in your classes as well. They're on-the-ground researchers and can provide all kinds of great advice.
That's actually a great point. A number of the undergraduates that have worked with us, my PhD students are all TAs. So when you're in a lab course, probably some of you have had some of my own PhD students supervising your lab projects as TAs. Get to know these TAs because they can give you an inside edge to get into a research lab.
Yeah, for sure. Okay, let's go to Benham's question in the chat. So back to the science. Was the cellulose research one of the difficult researchers to have it published, or were there similar cases in your previous research projects? Like, is that a typical path for publication?
Yeah, another really good question. So as I said, we got into this completely by accident, and it's entirely unrelated to anything I had done before. So when we wanted to publish this, I didn't know any of the researchers in the field. And when it goes out for peer review, if the people don't know you, then it's a lot harder to get it published than if you've met them before at conferences or they're aware of your work. So when we submitted our first paper, we submitted it to the top paper in all of science, which is Nature. And we got 2 referee reports, one that was glowing and one that was quite negative. And we had to go back and forth for quite a while on the negative one before we were able to get it published. And then but the editor was on our side. They clearly wanted to publish this in Nature as long as we could resolve a few of the scientific issues that were being raised. Some of the papers that followed, I would say the next 5 papers or so that followed, we had a really tough time publishing because we didn't have any reputation in this field. Now it's much easier for us because everybody working with cellulose nanocrystals is aware of, or not, I shouldn't say everybody, but a lot of people are aware of our group's work.
Okay, let's go to William's question. This is a good question as well. So many good questions from our students. So William's asking, you mentioned that when you were developing a chiral pneumatic elastomer, you had to swell the film to enlarge the helical pitch. Could you shrink the helical pitch beyond the point that it reflects visible light such that it reflects UV radiation? And if so, what sort of applications could this have? And he says, thank you for your time.
Yeah, so we can make materials that, like we can make the glass material, for example, that has a helical pitch that's very short so that it reflects infrared light, or sorry, it reflects UV light. These could be used in coatings on eyeglasses or sunglasses. Could also be used for, glass coatings on paintings to remove photo bleaching and other problems that they get from exposure to UV light. We're really interested in the ones that have longer helical pitches and reflect infrared light, because we can make colorless films that reflect infrared light and could be used to reduce the amount of heat energy that gets into a building, and that would reduce the amount of air conditioning required to keep the building at a reasonable temperature.
Oh, there's so many interesting applications. I had a question. I'm just going to sneak one in before we get to Adrian's question. With the elastomers, could they be embedded in like structural materials for like sensing, road decay or bridges or things like that? Like, just dawned on me.
Great thought. One of the applications that we were looking at was putting these onto structures like bridges to detect small earthquakes or damage from earthquakes.
Yeah. Okay. Yeah. Interesting.
And one thing I should say too is I showed you stretching them and they change color, but you can also press them and they change color. So they could be used for finger sensitive displays and things like that.
Oh, neat. So yeah, and like I was thinking like wearable sensors or all kinds of cool applications.
Yeah.
That's really fascinating. Okay, so Adrian's question here, in your group, what is the research background of the people in your research group? Is it more organic chemistry, inorganic chemistry, physics, chemical engineers, etc.? What's the overall sort of disciplinary background or expertise?
Yeah. In my group, it's very diverse. The student who discovered the cellulose nanocrystal materials and published the Nature paper on the silica, his background was he had a biochemistry undergraduate degree from McGill. I have students, for example, let me see who here. Andrea is a student, she's a PhD student, and she joined me from Mexico, where she had a undergraduate degree in physics, but she was interested in doing materials chemistry. Kosei, his undergraduate degree is in chemical engineering, but he really wanted to get into materials chemistry, or actually he's doing synthetic organic chemistry now. It's kind of crazy. It's very different from what he did as an undergrad. Yoda is a visiting scientist from a company in Japan, and he's a fluorine chemist. So we've got a huge mixture of students with backgrounds in organic and inorganic chemistry as well as physics.
Excellent. Well, that might be a good place to end with that notion that it's okay if a journey kind of meanders and you might not know exactly what you want to study. You never know where your path may take you. So with that, I want to thank you, Dr. MacLachlan, for an awesome seminar. And thanks everyone for such great questions and engaging with us today.
Thanks again for having me.
Yeah, absolutely.
Yeah.
Thanks again. Appreciate it.